can start this nice symposium.
Yes, fantastic. Thank you so much. Thank you for that introduction, Hans. I hope everyone can hear me and see me. What I will actually do is I might actually stop my video just to ensure that we can get a good bandwidth on this as well, and we'll get going. So, fantastic. So, firstly, welcome. My name is Alexander All, and I'm a field application specialist or scientist here at ONI. And I'm super pleased to be here today with my colleague, Rebecca, who's, as Hans mentioned, is all the way over in California. It's an early morning for her there. And we're really pleased to be here to be hosted by SciLife Lab and work together with Hans and talk to you about the possibilities of super resolution microscopy. And as Hans mentioned, we're first going to do a bit of an introduction, and then after that, we will be following with a live demo of the Nanoimager, which is actually located here in the ONI headquarters in Oxford. So let's just dive straight into this. Let's see. So ONI is a biotech spin-out from the University of Oxford. And what we do is we make super resolution accessible so that we can help overcome some of the biggest challenges that we face within the biomedical research field. And what we're trying to do is to create accessible technology. And we've done this by building the world's first benchtop compatible super resolution microscope. And what we're trying to do is to produce clinically translatable results, because ultimately we believe that to accomplish this, we need to create a collaborative platform. And during my talk today, I just want to introduce super resolution technology and how it works, introduce to you the Nanoimager and its different imaging modalities, and couple this with a few different case studies of how our customers have applied our microscope within their daily research. So if we just start off briefly, the advent of conventional fluorescence microscopy really revolutionized how research was done within the life sciences. And it allowed scientists for the first time to tag particular proteins or structures with fluorescent labels to a very high degree of specificity. And so it was possible to visualize cellular components unlike previously. Unfortunately, the techniques that were used were limited in the resolution that they could achieve. And consequently, it created a barrier. Now, super resolution microscopy refers to a class of techniques that are actually able to break this barrier so that we can all of a sudden visualize with unprecedented detail cellular contexts at a single molecule level. So let me briefly explain the reason for this resolution limit. If we take just the single fluorescent molecule, such as a fluorescent protein that is in its entirety around 2.5 nanometers in size, when we actually look at this underneath the microscope, the light that we see that's emitted from this fluorophore travels through the objective lens and it diffracts. What that means is that what we actually see is a large blurry spot. And this spot is at best somewhere around 250 nanometers in diameter. So it's almost 10 times the size of the original fluorescent protein. And this really highlights the issue with conventional fluorescence imaging, in that the structure that you're trying to see in its entirety might be smaller than 250 nanometers, but the light that you're detecting from just that single fluorophore is larger than the structure itself. Not only this, but if you have multiple fluorescent proteins all within close proximity to each other, what we're going to start seeing is a convergence of signal. Now, what that means is that there's no way to actually distinguish between these fluorophores, and we can't actually spatially resolve our markers. So how do we get to a point where we can actually resolve between them? Well, if we go back to just the single fluorophore, there are a number of ways in which we can accomplish super res. The first is called structured illumination microscopy, or SIM. Now, in the classical SIM approach, we use a patterned illumination, and usually stripes are placed within the excitation beam path. Now, these stripes are positioned and then changed a number of different times, and the emitted signal is then recorded continuously in different positions. Now, SIM is based upon this interaction between the structured excitation and the samples. And what it does, it creates what's called a Mornet pattern. And this Mornet pattern contains high frequency spatial information that can't be resolved in diffraction limited imaging. So we need to process these images using algorithms, and then we can reconstruct to produce a final image that has a resolution somewhere around 100 nanometers in the X and Y plane. Now, another technique is called stimulated emission depletion microscopy, or STED. Now, STED is based on two lasers that spatially pattern the diffraction limited spot. And when used together, we create first the excitation laser that excites the fluorophore, and then we have an overlapping depletion laser that creates a donut-shaped ring over the top. And what this does is it removes signal from the periphery so that we're only illuminating the center of the actual diffraction limited spot. And what we can then do is use the single photon detector to pick up the single signal during an acquisition. Now, STED has a resolution limit somewhere around 30 to 70 nanometers for commercial systems, but it also has less, and it can be dependent upon the size of what we choose the donut-shaped hole to be. Now, the final technique that I'd like to talk to you today about is single molecule localization microscopy, or SMLM. Now, this is based on the notion that diffraction of light occurs in a predictable manner, and it's dependent on the numerical aperture of the objective lens, as well as the wavelength of light from the fluorescent molecule, which means that what we can do is fit a Gaussian to this. And then we can very accurately use an algorithm to localize the actual position of the single molecule within the diffraction limited spot. So this is effectively how we overcome the resolution barrier. And it basically forms the entire basis of how the nanoimager uses a combination of techniques to acquire a super resolution image. So for the rest of my talk, I'm going to focus on single molecule localization. Now, importantly though, this technique only works if it's possible to detect a single diffraction limited spot in isolation. And as we know, normally in your cells or your tissues, all the fluorophores from the labeling will be emitting light simultaneously once you turn on the laser. So to isolate these diffraction limited spots, we use a technique which is called DStorm. Now, this stands for Direct Stochastic Optical Reconstruction Microscopy. It's a bit of a mouthful, really. But DStorm uses a very special class of fluorophore that under very high laser powers and certain conditions, stochastically or randomly, switch from a fluorescent state to a dark state, and then back to a fluorescent state again. And we term this behavior as blinking, very simply. So on the nanoimager, once we induce this blinking behavior by turning up the laser to a very high power, the software will automatically start to detect these single diffraction limited spots and predict the localization of the single molecule. Now, this happens over a large number of frames. We're talking somewhere in the order between 10 to 20,000 frames. And what we get is a super resolution image that is constructed point by point in real time. So with this in mind, I would now like to introduce the different imaging modalities of the nanoimager, and the first being DStorm and palm imaging. Now, palm imaging is very similar to DStorm. The only difference is that we're using specific photoactivatable fluorophores. So to illustrate the power of DStorm, in this example, what I'd like to talk about is nuclear pores. And these are actually the little gold structures that Hans has in his background, if we look closely on his virtual image here on the web chat. These ring structures are actually dispersed all over the nuclear envelope. And they're really important because what they do is regulate the transfer of materials in and out of the nucleus. And any form of dysfunction in this nuclear ring structure has been linked to a host of different cellular defects as well as diseases. Now, these pores are around 100 nanometers in diameter and lie well below the diffraction limit of conventional microscopy, which we can see here in the wide field comparison on the left-hand side. The DStorm acquisition gives us a much higher resolution, basically providing us an unparalleled ability to start to quantitatively explore our samples. And here we can use the integrated analytic tools of the nanoimager software called NIMOS, and we can now start to measure the diameter of these nuclear pores. So really, it allows us to design experiments that examine the characteristics of these nuclear pores and their distinguishing features. And it paves the way then for perturbation experiments so that we can study their conformation in health as well as disease. Now, the Nanoimager is not just limited to the single laser line. And on the Nanoimager, we can actually do multiplexed imaging. So this means that we can acquire D-Storm images in multiple colors. And it allows researchers to really examine markers and how they are spatially distributed in cellular contexts. So in this example here, we can see a dual-color D-Storm image of a mammalian cell line that's been transfected with two different ion channels. The BK, which is a potassium channel, as well as the calcium CAV13 channel. Now, it's been proposed that these two channels require each other for their activation. So one would hypothesize that there's some form of interaction. Now, both of these channels, in terms of their size, lie well below the diffraction limit. So we can see this from this wide field image, that we wouldn't have been able to understand this interaction without the ability to use D-Storm. So again, we can start to design experiments using this capability that will examine the distribution as well as the arrangement of individual channels. And this then gives us insights into receptor interactions as well as different entry routes that are taken. Now, the power of SuperRes really shines through when it comes to things like identifying interactions. And in this case, we're looking at a wide field image of the Phrococcus bacteria, which has been labeled with a cell mass deep red to visualize the membrane. Now, the outer membrane vesicle in this case has been labeled with a green Atto4A8 dye. Now, again, without SuperRes in this wide field image, it looks like the outer membrane vesicles are interacting together with the bacteria and some form of direct binding that occurs. However, if we then turn to D-Storm, we can actually start to consider this binding in a completely new light. And we can see that this isn't actually the case for the bacteria which is on the left. And we can confirm this by looking more specifically at the exact localization of points using the integrated software tools at our disposal. Now, I mentioned that the Nanoimager is capable of multiplexed imaging. And what that means is that we can cover, or rather we can take images using the 4 different laser lines that we have. And this means that we can image dual colors at the same time as well. So this is really useful for examining things like synaptic plasticity in neurons. Now, synapses have been identified as key regulators of neuronal disease onsets, and primary neurons in particular lie well below the diffraction limit. And there's been no ability to really examine their pre and post synapses within the synaptic cleft unless you turn to imaging these things using electron microscopy. Now, whilst electron microscopy can go down to a resolution of 0.5 nanometers, it doesn't really allow you to look more at the specific biomarkers. And therefore, electron microscopy images can be quite challenging to analyze. The 20 nanometer resolution capability of the Nanoimager allows us then to look at D-Storm with the 3 colors, and we can now start to differentiate the pre and the post synapses, as well as the synaptic vesicles that are moving between the two within the cleft, And this means that we can clearly start to understand how this technology is allowing us to be, or allowing neuronal research to become much more accessible when it comes to understanding and delineating the different progression of degenerative diseases and their own pathologies. Now, a really powerful example of D-Storm, and actually how the Nanoimager can provide a platform for therapeutic discovery, is in the identification and characterization of isolated structures. So in this case, we're talking about extracellular vesicles or viruses, as well as liposomes. And in this example, and in the coming slides, I'd really like to focus on extracellular vesicles, or EVs. Now, EVs are biological messengers that transport active molecules between cells. And their ability to be taken up by cells and alter gene expression makes them especially interesting for therapeutic purposes. However, studying them has been very limited because they are so small and lie well below the diffraction limit. On top of this, they're found typically in biofluids that are heterogeneous in nature, and they contain a host of different factors. And therefore, they require really elaborate purification steps to ensure successful isolations of these different subpopulations. So, following purification, we can actually fluorescently tag our EVs and then image them using D-Storm as a type of validation, and then examine the different biomarker distributions, Now, the nanoimager has itself quite a large field of view. It's 50 by 80 microns. So we can actually capture hundreds of EVs at a time, meaning that we can analyze quite a high throughput of data and could consider the different multi-factor characterization approaches. Now, if we look more closely at the DStorm imaging itself of these isolated extracellular vesicles, we can understand if the sample that we've purified contains aggregates, we can understand the different types of subpopulations, and we can understand the integrity of these EVs. What we're interested at this point is also, are our EVs intact? In this case, we're looking at the EVs that have been adhered to a glass surface, and they've been stained with wheat germ agglutinin, which is a non-specific membrane marker, as well as more specific tetraspanin markers, such as CD63 and CD81. And these are well-established markers within the extracellular vesicle field. This allows us then to consider compositions and to detect specific biomarkers and use DStorm as a quality control. Now, this is all well and good, but what we really want to know next is how many EVs are we looking at within our field of view, and how many EVs contain what compound. And this is where the integrative analytic tools of the nanoimager really comes to light, with our dedicated software tools to allow us to study the clusters of these different biomarkers. So now all of a sudden we can start to consider the particular parameters of the clusters that we're interested in and base that on localization numbers. We can look at area sizes, we can measure the densities of these. And the best part is then we can save these constraints or filters and we can apply them across multiple data sets. This allows us then to consider the spatial distributions as well as the arrangements using established algorithms. Importantly, what we're creating here is a really friendly user interface, meaning that we can generate results and worry less about the complex mathematics that is going on in the background. So with such a tool, the nanoimager really becomes a platform to study EVs like no other. Now, we've discussed a lot of DStorm and palm imaging, but the nanoimager isn't just restricted to fixed samples. It supports live cell imaging. Now, what I'd like to discuss is single particle tracking. And this works by tracing the diffusion of molecules in real time. And we can do this because the camera that the nanoimager uses isn't just highly sensitive, but also has a really high temporal resolution. And we can do 100 frames per second to generate really nice smooth tracks. And we're using the basis of single molecule localization technology then to study the dynamics and the behaviors of these particles. So what we can do is we can take this approach with the software and conduct the tracking within a solution of fluorescent molecules. And this is very similar then to the nanoparticle tracking analysis tools that are actually available out there. And what this means is that we can track the movement of particles through a solution of known viscosity and temperature, and we can then generate a size estimate. And this in effect works simply by larger particles will move more slowly through the solution, whereas smaller particles will move more rapidly. And we can then take the data and we can plot different types of distribution histograms. And then we can also start to identify the different peaks and understand whether what we're looking at in terms of the different subpopulations are what we are expecting to find. And this is really important again when it comes to conducting research on extracellular vesicles for the different diagnostic and therapeutic purposes. But the software tracking capabilities isn't just limited to isolated molecules. And the nanoimager itself, the body, can be heated. It can actually be heated up to 42 degrees Celsius. So what we can do is we can take this software and start to consider it in live cellular contexts. So in this example in particular, what the groups were trying to do here at the University of Edinburgh was to detect non-specific NHS-labeled EVs. And they were interested in vesicle dynamics following internalization in endothelial cells. So following imaging, the tracks were converted, and we can see them here on the very right, into this population network map, which have been colored by their coefficient deficient, which basically means how quickly they are moving. And we can then analyze this, and we can create really nice histograms to show this in a quantitative manner. One of the really interesting things that came out from this data was that we can see that there's a greater speed and distance from these EVs that is occurring at the cell periphery. And if we move closer towards the perinuclear region, there is a change in speed as well as distance. And this perhaps suggests a change in mechanism or some form of interaction that's occurring towards the nucleus. And in this experiment itself really paves the way for the next set of questions that we want to answer. Again, the dual color imaging capability of the nanoimager is not just limited to DStorm. We can actually apply it here, which means that we can look at this cellular structure with also the cellular context around it. So here is a beautiful example of single particle tracking that's been carried out in fibroblast cells. And here, the molecular tethering protein, PDZD8, is actually being tracked within the endoplasmic reticulum. Now, we can actually follow this on a population level, but we also have the ability to zoom in and look more precisely at how PDZ8 interacts with, in particular, membrane contact sites. And this allows us then to focus on these individual routes, and we can consider then potentially protein-protein interactions. And this in turn will allow us to understand how cell morphology might be regulated, as well as the cytoskeletal organization. Now, the ability to understand protein-protein interactions is becoming much more prevalent in today's research. And that brings us to the next imaging technique on the nanoimager, which is single molecule Fuston resonance energy transfer. Again, a bit of a mouthful to say, but what it is, in shortened, is just SM-FRET. And what it allows us to do is to study protein conformation dynamics. So for instance, what we can then start to consider is receptor-ligand interactions. So for instance, T-cell receptor and antigen interactions. We can study nucleic acid structure regulations. We can consider vesicle fusion, as well as forced induced conformational dynamics. So in particular, ion channel open-close properties. Now, the principle of FRET is based around very short working distances, somewhere between 1 to 10 nanometers. And in this schematic, we can see the principles on the very left side, whereby what we're seeing is if we have a green and a red probe pair, and we have a working distance which is beyond 10 nanometers, we're only going to see fluorescence from what's known as the donor molecule, so the green one in this case. But as they move within this tiny distance, so within 10 nanometers, what we're going to be seeing is a transfer of energy from the green to the red molecule. Now, in technical terms, this is due to a non-radiative transfer of energy. Now, the principle of this might be considered relatively straightforward, but measuring and quantifying such interactions is extremely challenging. Now, since the nanoimager originally came from a lab that specializes in single molecule FRET, it's actually been designed to encompass all the necessary tools to actually generate tracks in an automatic way. So for instance, what we can do is we can start to plot the individual tracks of both the donor molecule as well as the acceptor, or we can then consider the individual FRET traces and efficiencies. We can also look at this at an ensemble level. So in this example, what we did was we adhered DNA molecules to a glass surface, and then these DNA molecules were conjugated to Cy3 and Cy5 probes. Now in solution, they're going to be dynamic and moving around, but when they get within their working range of 10 nanometers, we're going to see a change in fluorescence from the green to the red. And as I mentioned, this then is automatically detected within the software so that we can perform specific analyses. And the best part is because of the large field of view, we can really try and capture thousands of these events occurring at a time. So now I've discussed the different varying imaging modalities so far that are available on the nanoimager. And for the next couple of slides, I'd really like to present a case study from the bacteria field using the nanoimager. Now, this was done at a group at Imperial in London, and what they were doing were examining adaptive responses. Now, bacteria spend most of their lifetime in a state of no to little growth, and therefore they need to adjust their gene expression to attenuate and reduce their metabolism. Now, this is because they obviously require different types of essential elements, such as carbon, transition metals, as well as nitrogen for their survival. So the researchers in this case were interested in the bacterial mRNA binding protein called HFQ, which is known to be involved in spatiotemporal regulation of gene expression during periods of stress. So HFQ was tagged with a photoconvertible M-cherry fluorophore, and the bacteria were then stressed, and they were stressed by inducing nitrogen deficiency over a 24-hour period. and then they were imaged using Palm single particle tracking. Now, it turned out that there were large accumulations of HFQ that gathered into one large foci within each of the bacterium. Now, this wasn't apparent within the wide field imaging, but we could see that much more clearly when we turned to the Palm SPT. So what the group were really interested in trying to see were the dynamics of the individual HFQ molecules. And what they did was, following the single particle tracking, they turned to the software, which was able then to map out the diffusion coefficients, firstly, obviously, for the entire field of view, but then they could also do it for the individual bacterium. And we can see quite clearly that the movement of HFQ is significantly less and mostly static within the nitrogen-starved bacteria. But in comparison to the control, the HFQ dynamics were much more at a baseline, and they were moving much quicker. Interestingly enough, when nitrogen was actually added back into the bacterium, these foci completely dispersed within 2 hours, and these foci turned out to be completely reversible. But let's say we wanted to explore these foci further and compare them with our control, for instance. What if we wanted to explore them in multiple acquisitions? Well, if we then go back to our Palm data set and we start analyzing it using the cluster tools that I talked about previously in our EV data set, we're actually explicitly able to define the parameters to understand the metrics of what these foci are. So what we can do is we can start to consider the different area sizes, we can start to consider their densities and localization numbers. And once we have all these parameters, we can then quickly apply them across different data sets, because what we have is a defined signature that we can apply elsewhere. Now, I've talked a lot about the different imaging techniques so far. And the question at the moment is then, why is it that more scientists aren't using these techniques? Now, whilst I mentioned many of the benefits of super resolution, there are quite a few barriers to their daily use. And the first one being, they're usually huge. They're huge and they're complex, but they require incredible infrastructure. They require dark rooms, they require optical tables, they require cooling ventilation systems, and this all together makes them very expensive. Normally as well, they're dedicated to one particular techniques, so you'd actually need multiple systems to perform different types of analyses or experimental. Also, they're not very user-friendly, which basically means that we require very dedicated support staffs to run and maintain them over time. In comparison then, here's the NanoImager system. And the immediate feature that really stands out is its unique design. We've tried to create something here which is small. And we have, first of all, the microscope body, which is over here. We then have the actual computer itself, which runs the software. And then over here, we have the light engine, which contains the lasers, most of the electronics, as well as a cooling system. Now, because of this, the NanoImager is actually really well suited for live pathogen research. So together with that live cell compatibility, it means that it can be placed and operated safely within biosafety cabinets. And this is actually due to a few things. The first is that there are no oculars and there are no dials on the microscope, meaning that the entire casing is closed. Now, the best part of this is that we can operate this remotely, anywhere. And as you will see in a couple of minutes' time, when I hand over to Rebecca, that we will be running this remotely from another part of the world, another continent in that case. Now, this means that we can operate the system remotely and reduce the exposure time to biohazards. We can, on top of that, because of the way that we built it, it operates nicely in ambient conditions, and therefore we don't have to sit in the basement anymore, nor do we have to sit in these dark rooms. And it can actually be operated on a lab bench because it has enhanced stability and dampening materials that have been built into it. So, as we now approach the end of my part of this seminar, I'd just like to summarize with a few key messages. Firstly, single molecule localization microscopy on the NanoImager really enables us to consider different multiple imaging techniques on the single platform. The super resolution capabilities enables us to then to resolve structures down to the nanoscale. And couple that with the live cell capability means that we have multiple different experimental approaches so that we can track and map different protein dynamics. the dedicated software has all the tools that are necessary so that you can start your analysis immediately after the data acquisitions. And importantly, we've really tried to build an imaging platform that is for the user. Now, we really believe that the Nano Imager opens new and exciting possibilities within scientific research. And what we're really excited about is unlocking the power of all this technology together with you. So, without much more from me, and the moment that I guess we've all been waiting for, Rebecca is going to operate the Nano Imager in Oxford from her room in California. And thank you so much for listening to this talk. And if you have any further questions, please feel free to pop them into the chat box, and we're going to answer them as we can during, or sorry, after the actual demo itself. So thank you very much.
All right, thank you, Alex, for that excellent talk. Hopefully everyone can now see me, so I'm just getting my getting all my computers adjusted. So can everyone see my screen now? It should look like a bunch of blinking spots. Okay, cool. So basically, I'm gonna quickly run you through this and then tell you a little bit more about myself. So right now, what I've done is basically I've connected to a microscope in Oxford. It's actually, I think, next to Alex, or in the room next to him. I'm currently in San Diego, and so I'm operating this microscope remotely, which is pretty cool. Although it does require people in the lab in Oxford to put the sample on and put the buffer on and everything like that. So I've already started this DStorm acquisition because, as Alex mentioned, you do have to collect many frames to collect enough localizations. Sorry, I'm just, okay, fine. To collect enough localizations to build up a nice image. So right now I'm on frame 17,000, and sorry, I'm just trying to figure out where to put my zoom thing so that I can still see everything. Okay, and what we're looking at now are those blinking fluorophores, which are required to collect a DStorm image. So here, it might be hard to tell, but actually this is a mammalian cell that's been stained with an antibody against tubulin. So we're actually going to be looking at the microtubule cytoskeleton network. And what I've done is basically used all of the conditions necessary to get a DStorm image. So you can see here, I've applied a very high laser power, and this is to induce the fluorophore blinking. One of my colleagues has placed this sample in a DStorm buffer, which is an oxygen scavenging and reducing buffer. And we're using a fluorophore Alexa Fluor 647, which is a really common fluorophore used for DStorm imaging. It's actually the best fluorophore to use for DStorm imaging, and it's also a widely available commercial fluorophore. So that makes this whole process fairly simple. And if I zoom in, what you can see is the individual fluorophores blinking. And what the software is doing is it's basically capturing each one of those fluorophores. It's applying that Gaussian fit, which Alex was speaking about, and then the acquisition is complete. It's actually plotting it in real time on the analysis tab so that we can build up a super resolution image, which we can now see here. So I'm just going to go back to that acquire tab for a second and show you that blinking again. So basically, for each one of these localizations, if I zoom in, this is a diffraction limited spot, probably around 250 to 300 nanometers. But when the software detects that localization, it's able to find the center of it. And then that spot has basically a precision of around 20 nanometers or less. And so when it's plotted in the super resolution image, we now get enhanced resolution. So what I'm going to do now is basically show you a little bit of the analysis software. So with this kind of imaging, basically we want to make sure that all of those blinking spots, all of those localizations have a really high precision. And the way that we can do that is actually apply a set of filters to make sure that we are getting the highest resolution image possible. Okay, I think basically the image is still trying to save. So that's why the software doesn't want to cooperate with me. So you can see down here, it's actually still trying to save. So I'm going to actually just disable those again and move back to the Acquire tab. Okay, so everything has saved successfully, so that's great. So the first thing that we want to do is apply a drift correction. And what this is doing is, because we did have to take so many frames, this acquisition took approximately 10 minutes. I started it during Alex's seminar so that you wouldn't have to sit through a full 10-minute acquisition. But because it does take that amount of time, there's the possibility that there could be drift, lateral drift in the sample, which obviously we want to completely minimize. Any drift is basically going to blur the image, and because we're using super resolution imaging and we want to achieve as high of a resolution as possible, we don't want, we want to eliminate any of that drift. So we have a built-in feature which calculates the drift throughout the acquisition, and then you can apply that after the acquisition. And we also have on the, as that's processing, we have a feature as well, a focus camera, which basically does the same thing in the Z direction. So that basically keeps the correct Z plane so that you're not drifting up or down as the image is being acquired. The other important thing to consider to reduce drift is just to leave the sample on the microscope for a while to let it equilibrate to the temperature of the microscope and to make sure that everything's sort of stationary as you begin that acquisition. So usually what you'll see is in your first image or two images, if you're slightly impatient and you want to just get to the imaging, you'll see much more drift than if you wait for a longer period of time. So this sample has been on the microscope for quite some time now because we were, doing some testing before, making sure everything looks fine. So it doesn't look like there's too much drift in the sample, but it is good to make sure that we eliminate that anyway. So it looks like it's almost done. And then I'll go through some of the analysis tools. So we'll be able to look at filtering different localizations. So we'll be able to filter out localizations that have a poor precision or that have low photon counts, or we can even filter out certain frames of the acquisition just in case the blinking didn't look nice at the beginning or the end of the acquisition. I'll be able to show you how to measure the size of a microtubule. And then we'll also compare the D-Storm image to a wide field image so you can see really the level of detail that you're able to gain from that super resolution image. Okay, got it. So we've now applied the drift correction, which is excellent. And now we're going to go through those filters. So I'm just going to click disable all filters, and that brings up this menu of filters. And these are pretty standard. So first we have photon count. So that's basically filtering based off of the number of photons detected per blinking event. And when a higher number of photons are detected, you actually are able to obtain a better localization precision. So we want to filter for localizations or for blinking events that actually have a higher photon count. So I've just filtered, put the minimum at 1500 here. We can then also filter by actual localization precision. So typically I like to set this at 20 nanometers. but depending on your specific application, you might choose a different value for that. And depending on the quality of your DStorm image, you might choose a different value for that as well. The next is sigma, which describes basically the width of that blinking event. So you might remember when Alex showed you that diffraction limited pattern, and then he showed a Gaussian fit over that. So this sigma is describing the width of that Gaussian. And again, that's directly related to the localization precision that you can get out of the microscope. So I'm going to set that at 200 nanometers. The p-value describes the quality of the fit of each blink to the Gaussian. And then the frame index is exactly what it sounds like. It allows you to remove the first, either the first few frames or the last few frames. And this might be necessary basically because at the beginning of an acquisition, you're inducing the fluorophores to blink. And sometimes with a really high density sample, those blinks are actually overlapping, and it's very difficult to detect them and fit them if they're overlapping. So you might want to filter out the first few hundred or thousand frames. So I'm just going to filter out 1,000 frames here. I don't really think it's necessary for this image, but just to make sure that we're only getting single blinking events. Okay, so now we can see a very nice super resolution image. Again, this is looking at the microtubules network. And if I zoom in, we can really see that this image is built up of basically tiny points. So these points are those localizations that had been fit using that Gaussian algorithm. And just to give you a sense for size, we can actually measure the width of this microtubule. And we can see that, let me just make this bigger for you. This one is approximately 60 nanometers wide, which makes sense based off of the size of the microtubule and this resolution of the microscope of the method. And also, whenever you're doing DStorm imaging, you have to consider the fact that, especially if you're using antibody labeling, that your target is going to be a certain distance away from the fluorophore that you're using to image it, and that distance is going to be the length of the antibody. So you're actually going to add distance between your target and the fluorophore. Now, we can also measure the distance between microtubules. So for example, let me find a good spot. So for example, up here, we can see that there are quite a few microtubules that are actually crossing each other, and maybe we want to see the distance between them. So now I can draw that line again, and I can expand it out to cover these two locations. And now I can see, again, let me make this slightly bigger for you. The peak of one of the tubules is around minus 100, and the peak of the other one is about 75 or 100. So these two microtubules are about 200 nanometers apart. And this is, again, below the diffraction limit of light. So actually, if you were to look at the wide field image of this, you wouldn't be able to distinguish these two microtubules. And just to show you actually that that's true, I've loaded the wide field image that I took previous to the DStorm image. And you can actually see when we look at this location with the wide field image, it's really hard to tell what's happening at this intersection, right? We can see that there are several microtubules coming in, but it's actually unclear how many there are. Like here, is there one or two? Here, how are they crossing? It's just obviously a very blurry image. But if we turn on the DStorm image, now we can very clearly see that in this one location, which originally looked like it could be one microtubule, we can actually see two crossing each other. Here we can again see that in this location, there's actually more than two microtubules. There's 1, 2, 3. And here again, there's several at this intersection. So while the wide field, if I just zoom out for a second, you can see the wide field, oops, sorry. The wide field image actually looks pretty good when you zoom out. You can see individual microtubules, you can see their nice pattern and structure, but really the D-Storm imaging comes in when we're trying to look at those individual interactions between target proteins or between organelles, or even just looking at the quality of this cytoskeletal network. Okay, so I'll just show you a few other locations in the sample so you can get an idea of what this looks like. So for example, here, if we want to look at this intersection, again, we can draw that line histogram. Maybe we want to look at all 4 of these microtubules. We can look at it here. And we can again see all 4 of them. Actually, it's a little bit hard to tell because there aren't enough bins in this graph, so we can actually do this. And we can now see the spacing between all of those microtubules. And again, we can compare it to that wide field image and see that with the wide field, we really are losing all of that information. All right, so I think that's all from me for the live demo. I hope that that gave you a sense for actually how easy it is to take a D-Storm image. Like I said, you need basically 3 different components. You need the buffer, you need high laser power, and you need the correct fluorophore. But once you have those 3 things, it's quite simple. And with the Nano Imager, it's really simple to go from acquiring to the analysis. The localizations get plotted in real time, so you can already see that image building up as you are acquiring. Okay, so I'm going to stop sharing my screen now, and we can move to the question portion.
Okay, yeah, great. Thank you, Rebecca. So yeah, I basically hand over to you and Alex to start answering the question there. There was a first question about detecting RNA and DNA that you can maybe answer. And everybody that has question, either put them in the chat or raise your blue hand so then we can unmute you. So go ahead, Alex and Rebecca.
Yeah, sure. You want to go, Rebecca? Yeah, go for it.
Sure. So actually, the first question, I'll just read it aloud. Is it possible to get blinking labels for detecting DNA and RNA? And very nicely, one of our field application scientists at ONI, Siobhan, has actually already answered this question. So yes, you can use Yo-Yo or Cyto13, which both produce really nice blinking for D-Storm, and can be used for DNA and RNA. So that's quite convenient if you're trying to look at the nucleus or interaction between the DNA and RNA with other proteins as well. Okay, thank you for the next questions. I'll take the one, is it possible to localize an amino acid on or inside an animal cell? So basically with D-Storm imaging, you can image anything that you can attach a fluorophore to. So either using an antibody, so I guess if it's a single amino acid attached that's not incorporated into a protein, you would basically, if it's incorporated into a protein and there's an antibody against that particular protein, or if you can tag it with a halo tag, or even if you want to use a photoactivatable dye or protein and it can be tagged with one of those methods, then yes, you can visualize it using D-Storm or PALM imaging. basically anything that you can attach a fluorophore to, you can image with DStorm.
So I think the second one, is it possible to detect multiple targets on a single vesicle? So from that case, yes, it would be, as long as we can get them to create the actual blinking effect, which basically would mean that we have one that blinks and then that switches off, and then another one would fluoresce, so that we would be able to distinguish between those two diffraction limited spots on the single vesicle. So then in that case, the other thing is that we can change the different, one thing that we didn't talk about was changing the illumination angle, which allows us then to create better signal to noise ratios. So the nano imager actually has the capability to go from the epifluorescence, where it's going straight through the laser, or we can actually angle it all the way down to a turf angle. But we have the ability to control it ourselves. So we can actually do anything in between, which allows us then to do a technique which is called high-low imaging as well. So that's how we can regulate to get better signal to noise ratios.
Yeah, and like Alex was saying, so all of the data that he showed with the EVs, the extracellular vesicles, that was looking at multiple targets on a single vesicle, in this case an extracellular vesicle. And so using different antibodies or tags against different proteins and different colors associated with them, you can image multiple targets. So he was showing, I think, some 3 color images, so 3 targets on a single extracellular vesicle.
Yeah. The 3D information, so someone asked, can you get 3D information, for instance, using a biplane imaging? So the nano imager does support 3D imaging, and we're able to do that using an astigmatized lens, which basically creates these diffusion or diffraction spots, in an ellipsoidal manner, and we can actually figure out their length in the Z plane by doing that. And then we can create that on, we can extrapolate that data so that we can get a 3D image in DStorm or palm imaging like that.
Okay, so there is a question about how we prepare our cover glass or special microfluidic systems. So basically, when you're doing DStorm, especially since we're using a 100x objective and we're using typically turf illumination, we always need to use cover slip thickness glass, so 1.5H. And we clean it thoroughly. There are lots of different cleaning methods that you can use that you can find online. Everyone has their particular cleaning method that they like, but it needs to be it needs to be cleaned so that you basically remove any organic matter from the surface. And then it really depends on your application what you coat the cover slip with. So sometimes, you know, a very common one is to use PLL, PLL coated cover slips, which just helps the cells to adhere. You might also use just BSA, or you might not need to coat with anything at all. So that really depends on your specific application.
So yeah, the nano imager does support the Z stacking possibility, although we, for DStorm acquisition, it might not be very applicable from that sense, but it is possible to do something similar. In terms of also, I should just add with the DStorm far red, there's definitely other compatible ones. So we can choose the different wavelengths all the way down. We use Alexa fluorophores, which we find are very good at the 647 range, but you can go all the way down to, for instance, you can use 488, but we would recommend using something like Atto 488 in that case. So there is a commercially available, there are a wide range of commercially available ones that you can use. And then just to add on as well, so you can use things like Click, you can use snap tags, as well as halo tagging, which are becoming increasingly popular, so that you can specifically label different types of fluorophores.
And just to add on to what Alex said about the Z-Stack imaging, so basically this microscope was made to be a DStorm super resolution microscope, but obviously as soon as you get into that kind of imaging, it's really useful to have, you know, features like Z-Stack, time lapse, multi-acquisition, so that you can start asking questions about live cells as well and perform the single particle tracking experiment. So on the Nano Imager, you can perform Z-stacks, you can perform time lapses, and we have a feature as well called an overview scan, which basically scans a certain field of the sample, and it's an interactive map and basically allows you to find cells of interest and specifically image those cells. So it's sort of our way of getting a 20x field of view with a 100x objective.
So that last question with the software and how you can actually see. So once you start doing your filtering analyses that Rebecca was showing you earlier, what you can do actually is once you start filtering, it will show you how you filtered and the different fluorophores and the statistics that are not being counted within once you've actually done that filtering. So you are able to see the different changes that you are manipulating as well.
And to answer the question about flow chip models, so the microscope basically has 2 openings. It actually on the outside has no buttons, but the microscope itself does have 2 openings. One is where you place the sample, where the objective and the stage is, and then another one is in the back. And you can use that access to the microscope to basically add in microfluidics or to add flow, CO2 for buffering the system for live cell imaging, or for basically accessing the sample where you need to be able to access it as the microscope is running. So that allows all the lasers to be able to stay on, but for you to reach the sample in a different way.
Yeah, so just on that, with the live cell imaging, it's possible to use things like a pre-buffered CO2 medium or something like that, so that we can do short-term acquisitions and things. So something as well to consider.
And just to point out, Siobhan has also dropped a document in the chat with our table of popular fluorophores for DStorm. So these are ones that we've used and work really well in our hands. So we would definitely recommend those. Yeah, so for live cell imaging, so our laser powers basically can be, if you have a specific application for live cell imaging that requires low laser power, that's something that we can work with you to make sure that we're able to provide the right kind of power at the sample to reduce photo bleaching. But the other things that we can do, so we can use low laser power, we can also extend the imaging by using that time-lapse feature that I discussed before. Sorry. That I was talking about before, to put, to add time between the frames so that you can extend your period of imaging without bleaching the sample. And also, what was I going to say? We do have a very sensitive camera. So even when you're doing live cell imaging, you can use that low laser power and still get really nice signal to ensure that you can track the proteins of interest.
I do not think that currently we have a nano imager in Uppsala, no. But we would be very keen to get one into the Scandinavian countries at the moment.
All right, are there other questions?
So one of the benefits then of the remote system that we have with the Nano Imager is that in terms of support, it really comes down to designing a system which is almost a plug and play. As soon as you kind of plug it into the wall, you can get on and do your experiments. And the remote accessibility means that if anything were to happen as well, we have our customer support and our CX team, which are really able to go in and help you troubleshoot anything that might be of issue, which can really, you know, make sure that we don't have to be traveling back and forth. And especially during a time like this, with COVID ongoing, we have the ability to do a lot of remote support to ensure that you can access and move forward to troubleshoot anything that might occur.
Yes, and like Siobhan just added in the chat, of course, if you're interested in trying the Nano Imager with your own samples or seeing another more thorough demo, going through all the different features of the software, we'd be happy, you know, to be in contact with you and to help set up that that demo so that you can get a better sense for the microscope and how it can help with your research.
Yeah, absolutely. So in terms of a price estimate, this is something that we would be discussing together with you. So yeah, in terms of with everything included, the way that the system works, it would be priced somewhere around 245,000 euros as the start. So yeah, and yeah, thank you, Siobhan, for adding in. So this is also including obviously the software and the installation and all the training. So everything is basically baked into one price from that. So in terms of cost per samples and things, so in terms of arranging demos, that's just a matter of us communicating, and we can easily set up some specific chats, and then we can ensure that we test your samples together with you, and we can perform live demos, and we can go from there to ensure that you know, we validate to ensure that the system works for you. Yeah, I don't know if there are any more questions. Hans, do you have anything that you'd like to add or end with, or anything like that?
No, well, not really. Oh, as you already said there, we don't have the only platform around yet here in this part of the world. But as Rebecca also said, it can be remotely pilot tested. If somebody needs support with super resolution microscopy, you can of course contact me at SciLife Lab, and we can help you with the techniques that Alex mentioned in his talk there, the SIM and the STED, and we can also do single molecule localization microscopy support. So now I don't think I have anything more than, yeah, the purpose was of course to advance the knowledge about this technique, and I think, and I hope, yeah, we did so. So I think we can close the webinar. And people can of course get in contact too, and I by contacting you or Rebecca and everybody, I guess.
Yeah, absolutely. So you're very welcome to contact me or Rebecca, and yeah, we can obviously take it from there. So our website, you're also very welcome to visit, and I will send out more information, and we can happily discuss things. Yeah, not a problem.
Yeah, maybe you can drop your emails in the chat so people
absolutely.
can copy them right away.
I'll put that right here. Yeah, and if you have any more questions, I think if you have any more questions, then yeah, just please feel free to email us, or we'll get in touch with you. Thank you very much for attending.
Yes, thanks for giving a nice talk. Okay, yeah, bye-bye. Take care. Stay healthy.
Thanks,
Bye.
everyone.
Yeah.
Keep safe.
It was
Bye-bye.
fun.
Waiting for, yeah, I think everything worked fine, or? I think everything worked fine, or what do you think, Alex?
Hey.
Hey.
Yeah. No.
Yeah.
I think it worked right there.
Yeah.
Yeah. No. Early morning for you, Rebecca. You're all woken up now.
Oh, yeah. Yeah, I got up at 4.
Yeah. Cool.
But yeah, it was really good. It was super fun.
Yeah. That's good.
Yeah. No, I think people liked it, as they also wrote in the chat, and they were eager to get hold of the machine and also get support for different projects and things like that. So
yeah, they seem to be eager to get their hands on it, which is a good sign. At the end of the day, it's one of those things that we really like to make sure that we push, right, is getting people, you know, quickly onto these systems, which is
yeah, exactly. Yeah, it's been a hurdle to get people going into these directions.
yeah.
So.
Exactly. No, so no, I'm really pleased. I'm glad that we're here and we've managed to carry this out. So
Yeah.
but yeah.
Okay. Yeah. Thanks then.
No, thank you very much.
Keep in touch.
Yeah, absolutely. So what I'll do is I'm gonna send out a bit of a thank you email to everyone who has been attending, and then we're gonna go from there. But absolutely, Hans, I will send you an email later today and catch up and
okay.
from there.
Yeah, cool. Okay, take care. See you.
Cool. Thank you very much.
Bye. Bye.